Hello everyone. In this episode, I would like to share with you how to use the BrainCog platform to construct a mammalian thalamocortical column based on anatomic data. The functional minicolumn is an essential structure of the brain cortex. Many cognitive functions in the brain depend on this structure to be realized. So the research on the structure and the neural activities of the functional minicolumn is very important. In the platform, this structure is based on the anatomical data of the mammalian brain cortex. The data mainly refers to the article published by Corticothalamic in 2008, which released detailed mammalian thalamocortical anatomic data. This data is derived from the Anatomy of Cat Area 17, which contains 22 types of neurons, including 8 types of excitatory neurons and 9 types of inhibitory neurons in the cortex, 2 types of excitatory neurons, and 3 types of inhibitory neurons in the thalamus. At the same time, the firing patterns of these neurons with different morphological characteristics are also different. Here, we still refer to the Corticothalamic neural model to simulate the firing patterns of these different neuron types. Neural circuits are also realized through the interconnection of different neurons. Since the cortex consists of 6 layers, different types of neurons are distributed in these layers, and a large number of dendrites and synapses will also be distributed in these layers. For example, in the 4th layer of the cortex, it's mainly distributed with a large number of stellate neurons, and their synapses project to neurons in other layers. At the same time, there are numbers of synaptic connections between the cortex and the thalamus. The synaptic connections architecture is summarized in this table. This table is so detailed that we can construct the structure of the minicolumn directly. This video shows the entire structure of the minicolumn. There are 6 layers in this column, and we stimulate the stellate neurons in layer 4. In layer 4, they will be triggered and emit spikes to other neurons they connected. And we can see the activities of the neurons when the column is unfolded. Yes. Okay. Next, let me show you how to simulate this structure in our BrainCog platform. First, you should open the BrainCog folder and find examples and multi-scale brain structure simulation, and open this corticothalamic column. Yes, in this folder we can find the main.py file. Open this, and you can run this file to get the results. You can see that the neuron numbers are different in different cortical layers, and if the column contains 1,000 neurons, the number of synapses can be reached to 4,020,000. There are some important functions to be noticed. First, we can set the total number of neurons in this column. Here we set 1,000. You can try 10,000 architecture. Next, this function. Create cortex network. This function is responsible for constructing the whole structure of the column. We can set some features of synapses and cortical layers and dendrites and thalamus. And the most important thing we should notice is that we use each curvature model to simulate these neurons, and this model inherits the basic class. Here, the basic class. Here. The basic class of the BrainCog, and we add more parameters here, and about the details of these parameters, you can refer to the CSV files in the folder tools. Yes, all the important data can be found here. Yes, when we after we construct the structure of the Minicolumn, we can find, yes, we can run this Minicolumn. You can find this function here. We can find the neurons in layer 4 and input the current. Yes, layer 4, and input the current to these neurons, and we can observe. You can see this. Yes. We can observe the neurons, the activities of the neurons of the entire Minicolumn. Yes. Okay. That's all for this episode. If you are interested in our research, you can download the BrainCog and run it. For more information, or if you have any questions, please email us. Thank you very much. Best wishes for you.